I'm so honored to be here, and I want to thank the steering committee. I want to thank Lorraine and Emilio and Louis and Vicky, and thank the people of this land. This may be one of the smallest audiences that I've spoken in front of, but this is probably the most significant, and I say that with great sincerity. A recent quote from a Native American elder, and I wish I knew the name, so if anyone knows who spoke these words, I want to give proper credit. But he said that we do not inherit our environment from our ancestors. We borrow it from our descendants. And I think that is so true. In the next hour and 20 minutes, I'm going to show you some things that I think are self-evident truths. And nature is unbelievable in this complexity of design and successes and creations. I am, in a sense, rediscovering that which our elders long ago discovered, that mushrooms are deep reservoirs of very important medicines. And I stand here, and I wear my favorite hat. This is my hat. It makes me look rather dapper, and I'm balding on top, so it keeps me warm. And it is made from this mushroom, which is called amadou. The Latin binomial is called Fomes fomentarius. Amadou is one of the most important entities that humans associated with for human survival. Indeed, it can be said that a lot of us here today are because of this mushroom. There's no doubt more than 100,000 years ago, we migrated from Africa. We traveled north, and we discovered something new in Europe called winter. Oops. This mushroom allowed for the portability of fire. You can hollow this mushroom out, put embers of fire inside, and carry fire for days. And as this audience knows in particular, the firekeeper of the clan may now hold a ceremonial role, but not too long ago, the role was extremely important for the survival of the clan. When this mushroom is boiled in water, it delaminates into mycelium, and you'll see lots of mycelium. And some ladies in Transylvania made this hat, and so this tradition goes back thousands of years into Europe. Now, this mushroom was first described by Hippocrates in 450 BC. It is the oldest known documented medicinal mushroom. No doubt, our ancestors were using these mushrooms for thousands of years prior to that. And I brought another fungal friend with me. And this is agarikon, the longest living mushroom in the world. It's resident in the old-growth forest of British Columbia, Washington, Oregon, and Northern California. It perilously survives in sky islands in Austria and Slovenia and northern Italy. Because the old-growth forests have been cut down, this species is now on the red list of extinction in Europe. This mushroom was described by Dioscorides in 65 AD as elixirium ad longum vitam, the elixir of long life. And I suggest to you today that this mushroom will be as important for human survival as amadou proved to be in the past. So I'm going to take you on a mycological mystery tour, which I'm really excited to show you because it's the culmination of not only my lifetime, but then that of many others who preceded me. This is the great earth that we live upon, and I believe that following into the future, if we follow the path of mycelium, we will lead to greater sustainability and greater biodiversity. I had an epiphany, and I like one of the waking moments. I like to lay in bed and just ponder the day. Just take 5 or 10 extra minutes and just think through the important issues that you need to consider. And this epiphany came to me out of the ether that habitats of humans share immune systems, and mushroom mycelia are the cellular bridges between the two. I sincerely believe this is true. I'd like to give respect to my teachers. These are my mentors, Dr. Alexander Smith, Dr. Daniel Stuntz, Kit Skates. These 3 individuals have now passed on, and Dr. Michael Bugh, who is my professor at the Evergreen State College. So rare is agarikon that in 40 years of going into the old-growth forest, Mike found his first agarikon this past fall. And this is a group of individuals, with the exception of Mike, but these individuals in today's political context you would consider to be conservatives, political conservatives. I met them when I was about 19 years of age, and they adopted me as their sort of mascot, their student. All the more surprising because when I was 19, this is what I looked like. Your suspicions are now confirmed, okay? So I'm going to progress through the historical record on the use of mushrooms. This is from northern Algeria, from the TASLC Plateau, which was called the Plateau of Running Rivers. Now, because of desertification, the Sahara Desert has encroached upon that environment. But about 5 of these pictographs were found in these labyrinthian cave complex in northern Algeria. And here's an example of what we call mycophobia, the irrational fear of fungi, which has actually permeated the scientific community as well. Even though there's 4 different peer-reviewed articles on the art of the TASLC Plateau that featured this image here, which has been redrawn by my friend Jonathan Meter, not a single scientist dared to suggest even, hypothetically, what the artist intended to describe. Obviously, the artist is very excited about mushrooms. And the association of the bee god and bee with mushrooms was an ancient method of preserving these mushrooms. And maybe it's not surprising that there is mycophobia, the fear of fungi, because in your encounter with plants and animals, your experiential window of that encounter expands weeks, months, years. Mushrooms are ephemeral, most of them. They come up just for a few days and they disappear. And to find mushrooms that are so short-lived in your viewscape of your life, that can feed you, that can kill you, that can send you on a spiritual journey, no wonder that we've lagged behind in our understanding of these mushrooms. But our ancestors knew a lot more, perhaps, about mushrooms than we do today. Another image 7,000 years ago in the cave. Clearly, this artist is communicating that the mushrooms are giving something to the mind, and I think this transcends time. And then this was discovered in Spain. This is a group of mushrooms that are associated with oxen, and this group of mushrooms here is thought to be putatively Psilocybes hispanica, a magic mushroom, an article which was published in the Journal of Ethnobotany specifically on this mushroom. And when we advance forward to about 300 to 400 years BC, this speaks to the origination myth of women of the seasons in Greek culture, where Demeter is giving Persephone a mushroom, and as Persephone consumes the mushroom, she's taken into the underworld. That's the beginning of winter, and then renewal upon spring, upon her coming from the underworld up to the earth, and then the spring and the seasons were reborn. This is the division between Austria and Italy. In 1991, a group of tourists discovered a body frozen in the ice, and because the glaciers were thawing, because of climate change, the body was uncovered, and these tourists got really excited. They found this frozen individual who obviously was very, very ancient, and they contacted, this is the border, and they contacted the Italian authorities, the border guards. And the border guards literally said, oh, another dead hiker, we'll come see him later. And these tourists realized, no, this body's thawing out, this is really important. So they went on to the Austrian side of the border, and they informed the Austrian officials, who came over and saw the body, and they said virtually, the father of the Austrian people, we found you. And they dragged him across the border, which created an international incident. And which has now he's been repatriated back to Italy. But this is Otzi, which is the best preserved remnants of a human that's ever been found in a non-funeral setting. So he died basically as he had a spear point in his back, and tethered to his right side with 2 of these polypore mushrooms, one of which was Piptoporus specialinus, a birch polypore, like this mushroom, and the other one was the same species. And here it is here. And this fabric that was tethered to his right side was the fire starter fabric that is made from this mushroom. Now, on our right side, for those of us who are right-handed, I have my knife, in this case, my laser pointer, and my car keys. We keep items of the greatest importance close at hand. And this position with the Iceman tethered to his right side was deemed to be significant because it's so important that he could keep fire and start fire. And he needed his fire starting tool kit to be very easily accessible. He needed to know where it was so he would not lose it, because to lose it could mean death. So I customized this presentation for this group here, but I thought this is important to bring out. This is some Anasazi petroglyphs that are extraordinarily unusual. And here are clearly, from my taxonomic viewpoint, these are mushrooms. When our peoples migrated over the land bridge in Alaska and came down, well, we had the knowledge of the resident organisms in our ecosystem. But then as the ecosystem became more arid, then we would lose many of these species. So this petroglyph has not been dated, but I think it must go back many, many thousands of years when these mushrooms were also resident within this ecosystem. And the Prince of Flowers, Cocopili in Mexico City, is adorned with many of the sacred plants. These are mushrooms. The incurled margin here is significant because this speaks specifically of a group of mushrooms, which are magic mushrooms called the psilocybin species, or otherwise known as psilocybes. This is Psilocybe hopii, a new species that has been discovered in the San Francisco mountain range north of Flagstaff, Arizona. And it is a species that's been named in honor of the Hopi people. So these mushrooms do and have grown in this area. And they are probably resident now in little islands, shrinking islands of ecosystems where the remnants of these species are likely to still exist. My wife and I spend a lot of time in the old growth forest. This is where we go to church on Sundays. And the old growth forest has become our large laboratory of experience. The old growth forest is something that I have a passionate interest and affection for. It draws me in and is a place that I use for my meditations. But when we look at an ecosystem, we now know something very, very different than science knew before, that all plants are part fungi. In Science, which is a journal, there was actually an article published a few years ago saying that all plants are part fungi. And so when you look at the cross-section of an ecosystem, there are the parasitic fungi that kill the trees, and then after the trees are killed, there can be saprophytic fungi that then rot the trees. And then there are the endophytic fungi, which are extraordinarily interesting. Several hundred endophytic fungi are growing parallel to and inside the tissue of plants. Scientists were perplexed when they do tissue culture in the laboratory of plants. They kept on getting fungi. They thought they were contaminants. We now know that these are endophytic fungi. They're mutualists. They help the plants survive. They create a host defensive resistance against pathogens. And that plurality and complexity of the fungal population existing within the plants and on the plants gives the plants to be able to resistance against disease and drought. And then the mycorrhizal fungi, an extraordinary group of species which can extend the root zones hundreds of times. The mycorrhizal fungi get sugar from the roots and the up-channel minerals. Here is a root that's coming near to the surface. As the mycelium reaches the surface, it exhales carbon dioxide, inhales oxygen. In fact, fungi respond to 4 primary environmental stimuli. When there's an influx of water, well, we all know that, mushrooms like water, but coincident with that, there's usually a drop of temperature as evaporation occurs and the clouds block the sun. So a influx of water, a drop of temperature, the mycelium comes to the surface, exhales oxygen, exhales carbon dioxide, inhales oxygen, and then they're exposed to light. The majority of mushrooms are photosensitive. They require light stimulation for them to form. So looking at the mycorrhizal fungi, they're spectacular in their ability that you can demonstrate the usefulness, something I very much encourage the community here to experiment with the mycorrhizal fungi. These are poppy seeds that were planted first into soil that did not have mycorrhizae, and the seeds here were dusted with mycorrhizal spores and planted side by side. One of my employees is a big skeptic. He did this in his backyard going, I don't believe this. And then he did this experiment on his own, and he's become a true believer. And an example also is all trees have these mycorrhizal fungi that also aid their growth. So when we look into the old growth forest, and we look at these hemlock trees that grow in nurse logs, scientists wondered how is it possible these trees could have enough light to be able to photosynthesize. When they took these hemlock trees and they put them into a greenhouse environment, gave the same amount of light that they were getting here in the old-growth forest, all the trees died. They wondered, how is it possible those trees could survive in the old-growth forest? Two scientists get credit, Arnbrandt, 1994, Simard, 1996. When they radioactively tagged carbon and nitrogen around the surrounding environment, they found something heretofore unknown in the field of science, that in the Populaceae, including aspens, cottonwoods, and alder trees, in this case, alder trees and aspens, has been shown, will move nutrients hundreds of feet in the sunlit riparian zones where these trees typically grow, and move it hundreds of feet to feed and nurture these small conifer trees. For the first time, it was known that mushroom mycelium bidirectionally budgets nutrients to many plants, including trees in the forest ecosystem, to support the biodiversity and the plurality of species within the ecosystem, because that biodiversity gave the ecosystem resiliency to catastrophia, to sudden changes. And so for the first time, we understood that the mycelium had a long-term view of the ecosystem to allocate resources so that complexity can be preserved. This is the largest mushroom in North America. It's called Brygioporus nobilismus. It weighs several hundred pounds. We've tried cultivating it many, many times. Prodigious producer of white spores. It weighs several hundred pounds. This species also grows exclusively in the old-growth forest. So the mycelium, when it grows, becomes tenacious. It holds soils together. The fungi are the grand molecular disassemblers of nature. They decompose plant and animal tissue, and in doing so, they build soil. So they are the soil magicians. But in the course of their growth, they produce these wonderful root-like structures called mycelium. And this form of mycelium is called a rhizomorph, a root form, because it's so tenaciously can hold the soil together. I did a measurement with a Mettler balance. Some of you will know what that is, a very sensitive balance. And I got a 30,000 times to 1. When I put 1 centimeter, weighed this 1 centimeter long, and then I weighed this, the ratio of the mass was 30,000 to 1. In fact, I could have made it 1/10 that long, a millimeter in length. It would have been 300,000 times to 1. So when the mycelium goes through the habitat, it holds it together, it grips it, it prevents erosion, but it does so much more than that. This is my manly man photograph. For 3 years, I was a logger. I set chokers in the Olympics. I was a long-haired hippie, as you saw at the time. I got enormous amounts of grief. The logging community can be extremely cruel, especially to new young men. And I survived, and not only I survived, but I was elevated to a key position. Then 3 of my teammates were killed in a logging accident. I was running Skylines. They wanted to elevate me at the age of 22 to be the leader of the entire crew. And I decided I'm going back to college. But looking at these trees when they're down, they do so much more than what many people had thought before. But in essence, the saprophytic fungi take wood. The wood becomes colonized with mycelium. Lots of things happen. Ultimately, soils being generated. Fungi create soils. And this is a soil horizon, not too far from where we live. Here's the clay layer. And this soil has been developed over 10,000 years. We live on an extremely thin film of soil. And the erosion of that soil causing the loss of biodiversity and is pulling apart the very food webs that would otherwise give us life. So these fungi play a critically important role for creating the very life support systems that are part of the food web. The mycelium then is triggered into mushroom formation. Again, a drop in temperature, influx of water, exhaling carbon dioxide, inhaling oxygen, and then exposure to light. Suddenly, the mycelium then concentrates into a little mushroom, and the mushroom then expands within a matter of 2 or 3 days. Mushrooms do not have a good immune system. The mycelium does. The mushrooms will rot, and they're only up for 4 or 5 days at most for most of these species. But like ripened fruit, they attract insects. And the ripened fruit then is quickly exploited by other members in the ecological community. Mushrooms, upon sporulation, upon giving off their offspring, then quickly rot. And the organisms that rot the mushrooms, we believe, or I believe, is absolutely critical for the downstream environments and setting the stage for rebounding the ecosystem that originally gave it life. So spores are being produced underneath the gills. This is a portobello species. Here's oyster mushrooms day 21, day 23, day 25. You can get 5 pounds of oyster mushrooms, wet weight, from 5 pounds of dry sawdust or straw. And then the mycelium is edible for the cows, chickens, and pigs. And it does so much more than that. But upon sporulation, these mushrooms are extremely perishable. And so as they begin to rot in the old growth forest here, and then they sporulate, lots of other things are happening, and then the mycelium goes subterranean. This is the majority of the life cycle of mushrooms, is at the mycelial state. It's like the tip of the proverbial biological or mycological iceberg. The mycelium is resident in the ecosystem for hundreds of years, and mushrooms only come up seasonally, and sometimes decades in between the times that you'll see them. In a single cubic inch of soil, there can be more than 8 miles of these cells. My foot covers about 300 miles of mycelium. Think of that. 300 miles of mycelium if you stretch these cells end upon end. I spent many years as a scanning electron microscopist. This is one of my scanning electron micrographs. And the mycelium, when it produces this web, is beautifully articulated, is a microfiltration membrane. So there's a flow of nutrients, like the rains that we just had yesterday, then is captured in this net. And the mycelium externally digests its nutrients. We know now from DNA analysis that we are more closely related to fungi than we are to any other kingdom. 650 million years ago, we split from fungi. In the Journal of Eukaryotic Microbiology, 4 years ago, 23 scientists published a new super kingdom called Apisthoconta, joining Animalia and fungi together, because we're more closely related to fungi than we are to any other kingdom, and our cells under the microscope look almost identical. We exhale carbon dioxide, we inhale oxygen, so do fungi. But when we look at this network, these little microcavities swell with water, so myceliated habitats hold water together. This is like mulch, what's full of mycelium can hold water together. And then as the rains go away and the dry season occurs, then these little cavities release water one at a time, like little bladders. And so you have sustained moisture over the long term because of the microcavities. And moreover, the pre-selection of the bacterial communities that grow within these cavities is determined by the mushroom species that is permeating throughout the ecosystem. So they set the stages for microbial successionism that leads to the plants that create the debris fields that then the fungi can feed upon. These are 2 movies I'm going to show you by my friend Patrick Hickey. First, there's territorial conquest. The mycelium grows out, and then it consolidates its control. This next movie has revolutionized our understanding of what's happening microscopically within the mycelial networks. The fungi produce bundles of nuclei called comet nuclei, and these hundreds of nuclei streams to the tips. And so the cells are normally binucleate. They have 2 nuclei per cell. But as they actively grow throughout an ecosystem, they become hypernucleate. And in a meter diameter patch of mycelium, there can be literally trillions and trillions of end branchings. And if there's a new insect, a new food, a new toxic waste that's never encountered in the evolution of these fungi, just think of these, the end branching being scientific experiments. If there's trillions and trillions of experiments, and one experiment is successful, and it generates a new enzyme, a new acid, a new strategy for gobbling up that potential food source, what happens? The mycelium streams. But some of that information via the nuclei back channels into the mother mycelium. The whole mat becomes educated. This is the concept and the foundation of epigenesis, which is a big buzzword now in biology, is the expression genetically of an organism in response to environmental stimuli. We have a lot of genetic expressions which are not expressed until we are challenged in nature. And so this has now made us much more clearly focused on the role of mycelium in nature and the fact that its network-like design allows it to create experiments that lead to the ability to adapt. So I postulated in the early 1990s that fungi were intelligent. I got a lot of accusations of people saying I was eating too many magic mushrooms, which I do not deny. But this group of scientists in Japan put the slime mold, Physarum polycephalum, into a maze, and the slime mold grew out randomly. They put 2 oat flakes at 5 exit points, 2 out of 5. Then the slime mold would grow through the maze, and then it would shut down the non-essential pathways. and grew directly to the sources of nutrition, disregarding any dead ends. Thus, they postulated this is a form of cellular intelligence. I would agree. But then the Japanese love doing things like this. And here's another Japanese group, more recently, in 2010, decided, well, if the slime molds are intelligent, how would they redesign the Japanese subway system? And so here is Tokyo, and here are the different satellite cities around Tokyo. They put the slime mold in the center, these all nutritional flakes that represent the sub-cities around Tokyo. They inoculated the slime mold, and it grew out, and you can see it grows out and makes connection to these food sources, and there's lots of other random branchings. In 26 hours, it shut down all the non-essential branches and redesigned the Japanese subway system in a more efficient manner than it is designed today. Moreover, when they applied mathematical analysis, the slime mold created a mathematical optimum. There is no better way of solving this problem than that which the slime mold had achieved. So if you have an engineering issue, you might want to consider consulting a slime mold. So I'm going to wax poetic here, but this is central to my belief system, and I don't think it's going to be strange to most people in this audience. The mycelium conforms to an archetype. That's similar to our brain neurons. This is a photomicrograph by Hank Morgan. And you look at the design of the mycelial network, it dawned on me that these looked like neurons, and the mycelial network conforms to the same archetype upon which our brains are based. And then looking at the computer internet, I was astonished when I saw this diagrammatic representation of the computer internet also conforms to the same architecture and the archetype of that of the mycelium. I believe the invention of the computer internet is an inevitable consequence of a previously proven evolutionary successful model. The Earth was bound to invent the computer internet at a time critical when information and resources need to be shared in order to prevent extinction. And so going further out, we're looking at the formation of dark matter. Most of you know that 95% of the matter in the universe cannot be seen. And the structure of dark matter also conforms to the mycelial archetype. And from Scientific American, the Deep Hubble Telescope view, this is 170,000 galaxies in this viewscape. These are the galaxies, and the cobweb of dark matter also conforms to the same archetype. I believe matter begets life, life becomes single cells become strings, strings branch to become membranes, and this is the way. We will find membrane, mycelial-like organisms throughout the cosmos. So let's go back in time. The first organisms that come to land were fungi more than 1 million years ago. Plants followed several hundred million years later. Fungi came to land first and associated with algae to form lichens. Most of you know that lichens are a mutualistic relationship between algae that can grow separately and fungi that can grow separately, but when they come together, they form lichens. And then the mycorrhizal fungi, the plants teamed up with the fungi to help them be able to march onto land. Well, the fungi are producing exotic chemicals in these extracellular droplets, and one group are oxalic acids, which are 2 carbon dioxide molecules joined together. Very interesting. And then they mineralize rocks. That's the term of art that's used. They actually pull minerals out of rocks. So they pull out calcium, iron, manganese, and this mineralization of rocks caused the rocks literally to be pockmarked. They have cavities. And so when it rains, the cavities fill up with water, so the rocks are no longer smooth. It leads to the crumbling of rocks and the formation of calcium oxalates. And the calcium oxalates are insoluble salts. So once these salts are formed, they sequester carbon dioxide. It's counterintuitive because fungi produce CO2, but they produce enormous amounts of carbon that's sequestered in the environment. And these salts then become resident in the ecosystem until enzymes or acids of another organism redissolves them. Working with Battelle Laboratories, we looked at these calcium oxalates, and our hypothesis is the calcium oxalates go off in advancing on an osmotic pressure wave in advance of the mycelium. And as they encounter a bacterium, for instance, as the enzymes of the bacterium cause these calcium oxalates to degenerate, the chemical scent trails then are picked up with the mycelium, which then tells the mycelium what is in advance of contact. And so the mycelium then through epigenesis is able to create a response mechanism that will make it more successful in combating potential adversaries. And I'm such an idiot. It took me forever to realize this. I would be tipping over rocks all the time and seeing mycelium. I never knew for the longest time that the mycelium actually was munching the rocks. The mycelium eat the rocks. And here's a clear example, the mycelium marching up into the rocks, pulling out these minerals and then channeling it to nearby plants. So this is how plants get the majority of their minerals, is through these mycorrhizal fungi. This fossil was discovered in 1859, laying down, and it grew between 420 to 380 million years ago. Now, at that time, 420 to 380 million years ago, that's before vascular plants, before flying insects. And so there were ferns, but there weren't trees, basically. And lying down 3 feet tall, 420 million years ago, this was the tallest organism on land on this planet. Well, it was a big mystery what this organism was. It was given the name Prototaxides. And for over 150 years, as a matter of scientific debate, until Dr. Kevin Boyce, a few years ago, in the Journal of Geology, published a series of articles and republished articles in response to the challengers. But we know now, de facto, proven by very good scientific method, that 420 million years ago, dotting across the face of the earth, were these giant fungi, over 30 feet in height. And they would attract lightning, of course. You know, lots of things would happen because of that. But think of that. Dotting across the earth were these giant mushrooms, 30 feet tall, before vascular plants, before flying insects. They would be a great reservoir, a great vessel for all sorts of other organisms to live within. So as we advance now forward from 380 million years ago to 250 million years ago, it was a time when all the continents were combined, at the time of Pangea. 250 million years ago, there was a great extinction event. More than 90% of the species in the fossil record, you can clearly see it, became extinct. And the extinction event was caused by potentially 3 different events. And it's a matter of scientific debate still. There could have been an asteroid impact that jettisoned enormous amounts of debris. In the fossil record, there's an iridium layer that can be found all over the world at one specific location 250 million years ago. There could have been volcanoes in Eurasia, or there could have been methane hydrate bursts from the ocean. I don't see these as being mutually exclusive. The asteroid impact could have created the earthquakes that caused the volcanoes to explode, that caused the methane hydrate to burst, but we know one thing for a fact. The extinction occurred, sunlight was choked out, and fungi inherited the earth. Those organisms that paired with fungi had a better chance of survival. We actually have the name of this fungus. It's called Reduviosporinites. That is evident in the fossil record, now extinct today, but this is the fungus that gobbled up the forests that quickly died. Then we advanced to the time of Gondwana 140 million years ago, when we have continental drift. And then 65 million years ago, we have another huge extinction event. This time we know what it is. It's not a matter of scientific dispute. An asteroid impact occurred. And 65 million years ago, it hit directly above where the Yucatan is. Enormous amounts of debris jettisoned into the atmosphere. We can see this again in the fossil record. Massive extinction. Sunlight is choked off, and fungi re-inherited the earth. There's a lesson here. As we are exposed to extinction events, those organisms that pair with fungi tend to survive. We've now entered into 6X, the 6th greatest extinction event known in the history of life on this planet. Dr. E.O. Wilson from Harvard estimates that more than 50% of the known species will become extinct in the next 100 years. We are probably losing species faster than we can identify them. And it's so important that we understand that for the first time, this extinction event is being caused by an organism, by us. We are not only the cause of this extinction event, but we're likely to be its victims. So I'm going to zoom down to, we live here in Washington State. Here's the Columbia River. And here is the largest organism in the world. And it is a honey mushroom that kills the trees. Armillaria astoi. It's an edible and choice mushroom, by the way. Kills the trees, but then the Forest Service wants to cut the trees down because of fire hazard. And we knew the GPS coordinates of this fungus. that was reported in the scientific literature. It is 2,200 acres in size, 1,665 football fields, and it's one cell wall thick. We have 5 or 6 skin cells that protect us from infection. The mycelium has 1. If you've ever done soil analysis, many of you have, it's not uncommon to find several 100 million organisms per gram of soil. That's a testimonial to healthy soil. And think the largest organism in the world is 2,200 acres in size and one cell wall thick. How is that possible? It is possible because of epigenesis, and these are self-learning membranes. They've learned strategies to be able to constantly combat adversaries and to grow to this huge expanse. Actually, Mark is a friend of mine. He had a small airplane. We flew down there. We couldn't find it. We went back. We looked at scientific literature. We had the long coordinates. We got in a plane again a week later. We flew down there, and we were right on top of it, but we couldn't see it because it was so big. So we spiraled up and up and up and up. We're in a tiny little cloth airplane, and we got to about 15,000 feet. And I've never been in a small airplane, without oxygen. And I told my pilot, Mark, I think I'm going to faint. He said, me too. That's not a good sign. So I said, well, let me get a photograph first. So I took this photograph, which is the best photograph of the largest organism in the world, I think, so far taken. But this is a lesson that we can learn from nature. My good buddy Louis talks about spirals. Well, that's so interesting because these mushrooms also form spirals. Spirals in the petri dish and spirals, in the woods as it gobbles up the trees. Why wouldn't it? It conforms to the basic laws of physics. The mushroom mycelium then is triggered into baby mushroom formation called primordia formation. And at the time of primordia formation, the tips of the mycelium become nuclear enriched. This is genetic material. Think of it as genetic intelligence. At the tips, at the vanguard of the fungus growing out, it's trying to have the best opportunities to be able to strategize through all the adversarial barriers that it otherwise would encounter. One of my electron micrographs can see the hyperaccumulation partially penetrated through the SEM beam here into the internal nuclei. The primordia then form, then mushrooms then burst. So powerful mushrooms are, they can break through asphalt. This is a shaggy mane mushroom. And we did a series of experiments, and I was working with Battelle Laboratories for a number of years, and breaking down oil using mushrooms. The Department of Transportation in Bellingham, Washington, had a yard where buses and trucks were stored for over 50 years. The Department of Ecology determined in Washington State that this was a toxic waste site, but they gave an exemption temporarily if the Department of Transportation could encourage and prove alternative environmental remediation methods. So there was 4 piles that were selected, absolutely similar. One pile was a control pile. One pile was inoculated with enzymes by a commercial company. Another pile was inoculated with acids, and then our pile was inoculated with mushroom mycelium. The mushroom mycelium absorbs the oil. The mycelium is white, actually becomes black in color. The mycelium then is breaking down the hydrocarbons of the oil and remanufacturing them into fungal carbohydrates. So these are breaking down, molecularly disassembling the carbon into elemental forms, and then they're repackaged as fungal sugars, the carbohydrates that build the cellular structure of these fungi. So we came back 6 weeks later. It was a big contest. The first pile, dead, dark, and stinky, had 20,000 parts per million of polycyclic aromatic hydrocarbons, 2% basically oil, very stinky stuff. You'd have to wash your hands with lava soap just to get the oil off your hands if you touched it. The second pile, dead, dark, and stinky, lifeless. Third pile, the same. And they came to our pile, it was covered with hundreds and hundreds of oyster mushrooms. And notice that the soil changed color. It went from black to white, which is a direct biomarker that the oil and the black was being metabolized and broken down. And some of these mushrooms became huge, a testimonial to the nutrition that they were able to capture from the oil. And so I call these happy mushrooms. They're extraordinarily large. And then, so our pile was teeming with life, and the other piles were dead, dark, and stinky. And then something else happened. The mushrooms would sporulate, they would attract insects, insects would lay eggs, eggs would develop larvae, birds would come in, bring out in seeds. And at the final, and unfortunately the camera didn't work, the final image, this entire pile was a green berm of life. The other piles were made dead, dark, and stinky, and our pile went from 20,000 parts per million of these hydrocarbons to less than 200 in 16 weeks. then approved by the Department of Ecology as being fully remediated, and then allowed for freeway landscaping. Now, the bacterial remediation companies and the phytoremediation companies were concerned. I like to build bridges, not burning them. That's why I love the concept of 4 bridges. This is, in a sense, the concept of 4 bridges right here. And so I said, well, mushrooms are the vanguard species. They break down the toxic barriers to allow the succession of other organisms. And so we're doing phytoremediation. The plants are now growing because they can survive that environment. The spores are attracting insects, and then after the insects are parasitized the mushrooms, then bacteria are growing on the mushrooms. So we have bacterial remediation. So the lesson here is that fungi lower the toxic thresholds that enable the downstream ecosystem to flourish with the biodiversity of life. So you can take an oyster mushroom kit bought from any company. It usually produces 3 or 4 crops or flushes of mushrooms 1 week apart. Then after that, there's not enough nutrition in the mushroom kit to be able to grow more mushrooms. You can take that expired mushroom kit, break it up, take crankcase from your car, pour it on the mycelium, and more mushrooms will grow. Absolutely phenomenal. We analyze the mushrooms. There's no hydrocarbons in the mushrooms whatsoever. But mushrooms hyperaccumulate heavy metals. And so we don't advocate people consuming these mushrooms from toxic waste sites because of the hyperaccumulation of heavy metals. We have disasters. We don't expect these disasters, but they do occur. We know we'll have a lot more in the future. But from the disasters to become opportunities for new solutions. So the Exxon Valdez, Alaska, the Costco Busan, and the BP oil spill all emitted enormous amounts of hydrocarbons and oil into the ocean. I had hundreds of people write me saying, Paul, what would you do? Because they heard about my experiments on land. I've never worked in saltwater before. So I called up several mycologists to ask them, would oyster mushrooms tolerate saltwater? They said, of course not, Paul. I go, how do you know? Did you try? They go, no, but it's obvious. I go, it's not obvious to me. And so I started doing some experiments, and I invented mycobooms. This is hemp socks filled with oyster mushroom mycelium on straw that float. They're only 75% moisture, though of course the water is 100%, so of course it's going to float. The specific gravity is different. And then we invented these mycobooms to corral the oil, and then the emitting enzymes then start breaking down the hydrocarbons, and then something else happened that was totally cool. This is in fresh water, this is in salt water. The alk acid in the mycelium would attract the flies, and the mycobooms then became a huge attractor for insects and fish. And so the fish at dusk would be coming up the mycobooms, going after the insects that were going after the mycelium. We have discovered in the past 2 years a revolutionary method that we're releasing to the public that reduces the cost of remediation with fungi by 100 times. It is the simplest things in life that sometimes are the hardest to see. And I can't believe it took me 30 years to discover this. And this is something we teach in our classes. And then we have a position paper that'll be coming up on the web soon on how to do this. So you don't need equipment. You can just use nature to be able to grow the mycelium. So you can corral the oil slicks together. There's no reason to clean up the mycobooms. They're biodegradable. And we can use the grasses all surrounding in the estuary environments as the material to grow the mycelium. Then we had Chernobyl occur. And Chernobyl created an enormous amount of radioactive fallout. The downstream environment was heavily polluted. And so a group of scientists were very concerned about the consumption of food by the Ukrainians. And so they went out into the forest ecosystem, testing all the wild potential foods and plants in the ecosystem to see if there was any danger for people to consume. Of course, there was great danger. But something amazing occurred from that analysis. They found one mushroom, the hyperaccumulated cesium-137, more than 10,000 times the background levels. So as the radioactive fallout fell onto the forest floor, the mycelium of this mushroom up-channeled the concentrated heavy metals, focused them into a mushroom, which became highly radioactive. But in doing so, decontaminated a wide swath of ground, thus allowing the plurality and the biodiversity of the ecosystem to survive, to give rise to the plants that ultimately would feed itself. These skill sets of these species speaks to mycodiversity, the importance of diversity. As we lose species and biodiversity, it's like losing rivets in an airplane. At what point will we go below the threshold where we have catastrophic failure and the ecosystem will unravel? Well, so the Chernobyl became a very exciting experiment. And then after Fukushima, my father was a nuclear scientist. He worked for Babcock and Wilcox. We had many arguments. I protested against nuclear power, made it very interesting dinner conversations. My dad was working for the nuclear power industry, and I was protesting, you can imagine. So I then had hundreds of people write me, what would you do directly after Fukushima? And so here's this mushroom that was discovered in Ukraine, the hyperculeate cesium. And I described within 5 days of the nuclear accident, used to sequester it and create a nuclear forest recovery zone. Don't try to greenwash this. This is a nuclear forest. And this was translated into Japanese, presented to the Fukushima legislature. And in the past 2 weeks, an enormous amount of momentum on this. I have gotten intelligence. I have several friends that are very high up in the U.S. intelligence service. And the Japanese government has been lying to the Japanese people. Up 50 to 100 miles away from Fukushima, there's more than 100 times the safe levels of cesium-137. They're moving 500 truckloads per day. The Fukushima reactor is heating up more now than before. They were venting once a week, now they're venting once a day. What do you think's in the steam? Is continual output of cesium-137. They're bringing in salt water from the ocean to keep the reactor vessels cool. And then they can't discharge that, so they have to store it. So it's a runaway reaction that's now producing xenon light. And so they had cameras there for the public to see, but physicists, universities are going, well, you shouldn't have xenon light. That means the reaction's running away. So then they flooded the vessels with white light. So you couldn't see the xenon light. This is a catastrophe, and their only solution is to scrape the soil all the way down to bedrock or volcanic rock and take it away. Fukushima is a great agricultural and forest area in Japan. It's a breadbasket of Japan. And as they throw away the soil, the breadbasket, is being destroyed as well. So this is a proposal that I made. They asked me, how long will this take? 10 years, 20 years, Paul? I said, no. This will take hundreds of years. 1 generation to pollute, more than 7 generations to clean it up. And so we have to do this in a piecemeal fashion where you harvest the mushrooms out of the radioactive environment, then you incinerate them, and then you capture the heavy metals, or you can use acids to dissolve the fungal tissue. And then the cesium-137 is put on epoxy, and then it's stored ad infinitum. But this is the best alternative that the Japanese people have seen in order to preserve their ecosystem and the biodiversity of the environment. If we can remove stepwise fashion the cesium-137, then the ecosystem will recover and be a good food growing area again. So after Chernobyl, a group of scientists were looking at videos inside the containment vessel of Chernobyl. 1 million rads of radioactivity, and they were astonished to see black molds growing all over the walls. On cement, folks, no other nutrition. 1 million rads of radioactivity, and these black molds were growing all over the containment vessels. They sampled the molds, and they found something heretofore unknown in science, that fungi use radiation for cellular metabolism in a fashion similar to how plants use light to photosynthesize. So this was a revolutionary discovery. It's unfortunate accidents, but from these accidents, we get new scientific discoveries. I've been involved in Google, and there's a sci-fi camp. They bring 140 scientists together, many Nobel laureates, and we all have a brain think tank. I met the leader of the Martian Project. 6 months to go to Mars, 1 year on the planet, 6 months to go back. I said, what's the biggest problem you encounter? He said, by far, food. We have to tow greenhouses in the vacuum of space. I go, how are you going to do that? It's so cold and all the energy. He goes, yeah, it's a huge problem. We can't carry the food. We have to grow it. This discovery here, that the ionizing radiation, both from the reactors on the space vessels and ambient radiation, in space, can be utilized for fungi when we can grow fungal foods. This accident and the subsequent discovery may be pivotal in the ability of humans to colonize outer space. Moreover, as you took your fungal foods with you, you could then terraform other planets because these fungi mineralize the rocks. So you could create the ecosystems and oasis environments that you could expand in the terraforming of other planets. So these disasters, as bad as they are opportunities for coming up with new ideas. This is where Dusty and I live. We have 20 acres on Skookum Inlet. This is the Squaxin Island tribe's land. And this is Totten Inlet and Skookum Inlet. 4 salmon runs a year, super rich in clams and oysters. And I bought this property in 1984, and I bought it, and 1 week later, the sheriff shows up. I thought that was awfully quick. I didn't even have time to do anything illegal. And every landowner on this inlet was served with a summons saying you had to replace your septic system within 2 years or you'd be physically pushed off the property. Because the E. coli coming from the uplands was harming the salmon runs, the steelhead, as well as the shellfish, the clams and oysters. I had cows, chickens, and pigs when I bought the property. 2 years later, they came 1 year later, they came back and said, Paul, we have an extraordinary thing happening on your property. What did you do? You have more than 100-fold reduction of coliform bacteria. And you have more cows, chickens, and pigs. All of us know they more than double in 1 year. And had not replaced my septic system. What I'd done is in the swales, I put wood chips. And I inoculated wood chips to mycelium. And the mycelium would hold the wood together, creating that web. And it created a microfiltration membrane and captured the bacteria. This was the dawning of the concept of mycofiltration, a term that I coined. And there's the mycelium, and these giant mushrooms would form. These are up to 5 pounds per specimen, which then would harvest larvae, which I fed the salmon, and then the fish then would eat the larvae that was hosted within the mushrooms. This was astonishingly exciting. Most of the bacteria are trying to kill the mycelium, but the mycelium preselects mutualistic bacteria that helps us host defensive resistant using the antibacterial compounds coming from the friendly bacteria to fight off the bacteria it doesn't want to have. So we have created much death. So this bioterrorism effort was a military successful strategy by the US and British governments. Gujal did not know how to identify Agarikon. Even though it's been resident in this culture, and this Argelic plague is from Charles Edenshaw from 1895, Gujal said we had 2 flu pandemics, so we had 2 smallpox pandemics. Each one of them wiped out 90% of our people every 10 or 20 years. We were disconnected from our ancestors because we lost that ancestral knowledge. So Dusty and I felt it was our role to bring back the knowledge of Agarikon back to the Haida and the Haida Gwaii. So we're at this overhang, and I think it's that the pictograph is the same that's in the back of the canoe. We don't know. But we're there, and we see this rock. We look at this rock. Now here's the rock, here's Agarikon, here's the rock, here's Agarikon, here's the rock, here's Agarikon. You get the picture here. And how likely is it we would find Agarikon where pictographs are speaking to the fungus man with the Haida culture? It was 1,000 to 1 to find it, and who knows, 10,000, 100,000 to 1. How likely is it we'd find a rock that looked like it's carved in the shape of Agarikon? And Agarikon was probably resident with those trees far before First Peoples ever discovered them. It's a sacred, shamanistically powerful place. Well, how likely is it we'd find it with a rock like this? I don't know. The numbers are astronomical. How likely is it we'd find it on my birthday? And this is when the photojournalist turned to one of our friends and said, does this happen to Paul and Dusty often? And my friends looked at him deadpan and said, yes. And this is where I think intuition and the discovery in science can be led if you are faithful, true to your heart, you respect nature, you respect indigenous knowledge and indigenous peoples, that you'll be led to these discoveries. It defies any form of scientific deductive reasoning. So we now today have 45 strains of Agarikon in culture, by far the largest library in the world. My lifetime goal is to get 100 of them. This requires us to go out in the old growth forest lots of times. And so now we are going into the old growth forest, and this is the one that I was holding up. This tree it came down, the area was logged. And this is on Cortez Island in British Columbia. And this is a friend of mine, he's a tree arborist and canopy expert, shot his guideline, and Scott Baker then went up this 700-year-old Douglas fir tree and found, and this is where the largest Agarikon, the longest living one in the world that we know of, grows. And very interestingly, it does not rot the branch that supports it. It calcifies it. Why would a fungus rot the branch that it wants to support it? It would fall. And so it ended up calcifying, like petrifying the limb. Extraordinarily clever idea. And so it's an epiphyte. Other mosses and other things grow on it, but this is the new growth. And so we have now gotten this strain in culture. We do not pick the mushrooms anymore unless it's going to be logged or there's a fire. And so this is the most recent one. Again, we were led to it. Amazing epiphany. This is in the upper Snoqualmie River watershed. And here's a young agarikon. Now we use GPS coordinates. We make GPS maps of it. And all we do is take a tiny bit of tissue with a stainless steel cork bore, leave the mushroom in the old growth forest, and then we take this amount of tissue, we grow it in culture, and here's an example of 4 strains of agarikon from the same island in the old growth forest. We match them up, and if they create this ridge of resistance, that means their strains are 2 different strains. And so these are different phenotypes. If they come together seamlessly, they're the same strain. So the diversity of strains is really important to us. Now here is an example of it's called a challenge test or a mating test. These are turkey tail cultures. You'll see these in a minute. And this is a turkey tail, 3 strains of turkey tail. These 2 strains, this is the one we used in a breast cancer clinical study I isolated 10 years ago. And then last year, Dusty found this turkey tail on our trail in the woods. I said, that looks really familiar. So I cultured both of them, matched them up. They grew together seamlessly. We found out that our land has become a fungal library. And so I was able to recapture the strain from nature, and it matched up perfectly, whereas another strain from another area would show the barriers of resistance. This is sort of my life. I do a lot of this. Then the extracellular metabolites, we already know they have antibacterial compounds. We already know they have compounds that break down toxic waste. And then the BioShield program contacted me after 9/11, and the biggest threat that the U.S. Defense Department identified by far is bioterrorism, because people could make this stuff in their kitchen or in their basement laboratory. And Earl Kern is a virologist, an expert in smallpox, and he said that any compounds tested have a 2 or more are active, 10 or more considered extremely active. We started testing our strains, and we're given the green light. The vast majority of 99% of the products we tested were pharmaceuticals. We didn't tell them that we had natural products. We just submitted them in water and ethanol. And we hit the big home run. We ended up having extremely strong activity against orthopoxes, and including smallpox. There is a National Public Radio interview, npr.org. You can listen to it, put in my name, Stamets and smallpox, and you can hear an interview with the Deputy Director of the FDA, the founder of the BioShield program, of the U.S. Defense Department, myself, talking about the importance of this discovery. We had a vetted press release. The vetted press release says more than 200,000 samples tested. We had the most powerful samples ever tested in the BioShield program against pox viruses. In fact, it's more than 2 million. I called up Colonel Huggins, he was my gatekeeper. The vetted press release had to go through the military. I said, well, do you test more than 1 million or less than 1 million? He goes, that's national security, I can't tell you. I go, well, more than 100,000 or less than 100,000? He goes, oh, way more than 100,000. More than 200,000. So it was funny. Somewhere between 200,000 and 1 million was national security, and I don't know where he drew the line. But you can read that press release also. It's vetted by the U.S. Defense Department. And then I was able to make the argument, which the U.S. Defense Department very bought into. Now, some of you may not understand the U.S. Defense Department is designed to prevent destruction of habitats now. They've had this big awakening that destruction of habitats leads to poverty, leads to disease, leads to terrorism. That connection now is very, very well understood. And that's a new thing that the U.S. Defense Department has realized. But I was able to make the argument that therefore, because this mushroom is exclusive in the old growth forest, we should save the old growth forest as a matter of national defense. And that argument has gone over extremely well. But smallpox is now in laboratories around the world. Hopefully, it's now extinct in nature. Those of you after 1968, you are not immunized against smallpox, so we were back to square one. But the big virus of concern by far is flu viruses. After Fukushima, or before Fukushima, I was watching 5 bird flu epidemics on the east coast of Japan. And the huge concern that H5N1, the bird flu, will become as communicable as H1N1 as it was 2 years ago, but have a 70 to 80% lethality. If you get bird flu, 70% probability you're going to die from it. But bird flu is not very contagious yet. But after Fukushima, an earthquake, a tsunami, a nuclear meltdown, what do you have? dead people, chickens, and birds, and pigs floating in a giant petri dish of salt water, exchanging their genetic material. Extinction events occur because of a confluence of characteristics. And we have entered into a period of viral storms. I was recently briefed, and a viral storm is mathematically probable in the next 10 years. It's coming. It's coming soon. It's going to change the very day-to-day activities throughout the entire world. A pandemic is on a near event horizon. And the fact that it hasn't occurred already is rather miraculous. So they started testing our samples that were active against smallpox, against other viruses, since they already had them. And so here's ribavirin, the positive control, pure pharmaceutical. Remember, anything that has 10 or greater is considered to be active. So here's the viruses, H5N1, H3N2, flu B viruses. Here's the positive control. And we gave them our little droplets that you saw from the mycelium, and water and ethanol. And here's our results. Now, we gave it to them in 35% ethanol, but we had to dilute it 100 to 1, because these are human cell assays, and alcohol is toxic. So 35% alcohol was diluted 10 to 1 to 3.5, 10 to 1 to 0.35. 100 to 1 dilution of our natural extracts of these old growth, of these fungi that grow in the old growth forest and elsewhere, and we beat the pants off of a pure pharmaceutical. This is the best results that they ever have seen, and this is something that I'm very passionate about. People ask me about patents. You know, I spent hundreds of thousands of dollars of my money on this. I'm up against the big pharmas. Do I want the pharmas to take this from me? No. So I filed patents on this discovery because this is novel, and I am creating hopefully a foundation. It'll be a nonprofit to spread this information and knowledge over the world. Viruses know no borders. They don't care about politics. So this is something that we should all be concerned about. And when you die of a viral infection, you die from bacterial pneumonia. It's the subsequent bacterial infection that scars your lungs that proliferates. So to find a mushroom that is dually active against bacteria, like Staph, E. coli, and tuberculosis, and is antiviral, is an amazing package. And when you go around, you go down and do what's called biogated fractionation. You can fractionate and the solvents, you can fractionate to an inactivity. By far, the natural product is a much more useful natural product for building host defensive immunity against viruses and pathogens than trying to find one isolated molecule, which is the pharma school. So viruses can cause cancer. Here are 5 viruses that we know that cause cancer: lymphoma from the Epstein-Barr virus, or EBV from herpes; liver cancer from Hep B and C; T-cell leukemia; cervical cancers cause HPV, and the Kaposi's sarcoma, and then Merkel cell carcinoma. So lots of viruses cause cancer. There's a lot more. And if you're immunologically compromised because of bad food, bad health, sedentary lifestyle, and you get a cold, and you get a cut, your immune system is taxed oftentimes beyond its limits. We all have cancer all the time. As your immune system cells are going around, natural killer cells and macrophages and T lymphocytes, they're scavenging them away. So it's really interesting to find a fungus that could be antiviral and activate the immune system and be antibacterial. 73% of all cancer drugs still currently have their origins in natural products. Don't let the pharmas fool you. Oh, just for the sake of time, I'll take 3 questions. So, yes?
Things I've gone up in the mountain. They're dry now.
Excellent. This is mine, May, and May has found a sister species to Agaricon. Agaricon's Latin name is called Fomitopsis officinalis. This mushroom is called Fomitopsis pinicola. Now, with the Kailuts, I believe I spoke to them, and they thought this mushroom was a sacred medicinal mushroom. It does also have antiviral properties. We tested it side by side with Agaricon, and Agaricon was much more potent. This one is about 1/20th the potency. But since this mushroom is potent, and since agaricon's so rare, it makes good sense that indigenous peoples would have found this mushroom also to make teas. So this is a medicinally important mushroom, and it's called Fomitopsis panicula. My wife argues with me, and she says it's pina cola, Paul, not panicula. But whatever you say, just say it with authority, and it seems to be good. So,
But
good.
this is not the turkey tail, this is
No,
more the
this is it's more like a reishi. It's
reishi.
more like a reishi.
Okay.
It's called Fomitopsis panicula. It's in my books, and you can look it up.
Okay.
Some interesting research. It's very strong in its anti-arthritic properties and antiviral properties. It's full of triterpenoids.
Anti-inflammatory.
Anti-inflammatory. So if you make some of this, it's 3 to 5 grams boiled in water in a tea, and drink the tea slowly over the course of a day. Some people have adverse reactions. About 1 to 2% of the population will have an adverse reaction. By drinking this tea very slowly over the course of a day, adverse reactions usually show up within 2 hours.
And may I share with them where I found it? I think when you walk through the forest here, I always look to see what's growing. So this was growing on some dead fir. And so it's pretty high up. And then this looks important. Thank you very much.
Okay, 2 more questions, and I want to show you a very short 2-minute movie that's took so much work. So yes, question please?
So we have a lot of PCBs in this area. Have you done any research with
Yes, I've been working with Jack Ward from Newfields, is a bioremediation company in Washington State, on PCBs, specifically around Mendocino County at the Georgia Pacific site where the sawmill is. PCBs are toxic in parts per billion. We've done our tests with microremediation, and I will tell you, as I call it, we were unsuccessful in breaking down PCBs because the enzymes are quickly attacking other molecules, and to get them trained to go after PCB and not go after all the readily available food has been a miss. We can get things that are parts per million, but parts per billion is 1,000 times less. And 10 to 25 parts per billion is toxic. So with the mycelium, when we do PCB in a petri dish, we can break it down. But once the PCB is in the soil, the mycelium has a hard time finding it. Okay? One more question? Yes?
What should we be growing here? We're so dry.
Well,
Of course, but it rains there.
well, Jim is here in the audience, and Jim, where are you? Okay, well, there's a pinyon oyster mushroom, and there's one that also grows on prickly pear, oyster mushrooms. It's very rare. I've never seen it. I don't know anybody who's ever found it. It's in the literature. So if you find a prickly pear with oyster mushroom coming out of it, let me know. And what we do for many, many peoples is that we go in, we'll isolate the cultures, we'll give them back their cultures, and we sign an agreement that we will never use these cultures for anything without their permission. Well, we offer that service, and we've been doing that with the macaws and the hideout and a whole bunch of other cultures in the Northwest. So if we could, I'm going to now show, I almost forgot this, and this is something that I think is important to see. And could we, mushroom mycelium represents rebirth, rejuvenation, regeneration. Fungi generate soil that gives life. The task that we face today is to understand the language of nature. My mission is to discover the language of nature of the fungal networks that communicate with the ecosystem. And I believe nature is intelligent. The fact that we lack the language skills to communicate with nature does not impugn the concept that nature is intelligent. It speaks to our inadequacy for communication. If we don't get our act together and come in commonality and understanding with the organisms that sustain us today, not only will we destroy those organisms, but we will destroy ourselves.
We need to have a paradigm shift in our consciousness. What will it take to achieve that? If I die trying, but I'm inadequate to the task to make a course change in the evolution of life on this planet, okay, I tried. The fact is, I tried. How many people are not trying? If you knew that every breath you took could save hundreds of lives into the future, how you walk down this path of knowledge, wouldn't you run down that path of knowledge as fast as you could? I believe nature is a force of good. Good is not only a concept, it is a spirit. And so hopefully the spirit of goodness will survive.
Thank you.
I guess you would say, oh. So, thank you, everybody, and peace.